Hello everyone, welcome to Lecture 4 Photosynthesis. Last time we were discussing the light-dependent reactions which occurred in the thylakoid membranes of the chloroplast. So we need to revisit just a couple things here, some of the products that were produced during this reaction. If you look here, you can see that water is being split, generating that oxygen gas molecule, which will be released, and you're getting a hydrogen ions in here. And these hydrogen ions are in high concentration in the thylakoid space. Outside, you also have hydrogen ions in the stroma, and as the electrons are passing through the electron transport system, the energy extracted from those high energy electrons was used to pump these hydrogen ions across the cell membrane, the thylakoid membrane there, to produce a further establish that hydrogen. So as those hydrogen ions pump across from the stroma into the thylakoid, you now further establish that concentration gradient, and now chemiosmosis, the second step here connected, is going to use those hydrogen ions, and these hydrogen ions, which are already in high concentration here, are going to pass through this special channel protein across the membrane. They're going to pass through this special channel protein called ATP synthase, and that protein is enzyme that is going to catalyze ATP production. The last thing we have being produced during the light-dependent reactions is this NADPH molecule. So when those electrons from photosystem 1 come up and get excited, they travel down, and NADP plus comes in, picks up the electrons. It also picks up two hydrogen ions fall along. One stays outside in the stroma area, the other one attaches onto the NADP plus, and you form NADPH. So it's this NADPH molecule and this ATP that are already out in the stroma here, this grayish area, and those are going to be used in the Calvin cycle. So now let's begin to look at the Calvin cycle, our next part of the biochemical pathway of photosynthesis. The Calvin cycle regenerates its starting material after the molecules enter and leave the cycle. So the cycle builds sugar from smaller molecules using ATP and the reducing power of electrons carried by NADPH. Carbon enters this cycle as carbon dioxide and leaves as a sugar. And the sugar here isn't just your typical glucose or whatever. It's called glyceraldehyde 3-phosphate, G3P or PGAL. The Calvin cycle has three phases. It has the carbon fixation phase, which is catalyzed by an enzyme called rubisco, and then you have a reduction state, and then you have the regeneration of carbon dioxide acceptor, RuBP, ribulose biphosphate. So if you look here, here is the first step, the initial step of the Calvin cycle, where you have this three molecules of ribulose bisphosphate, RuBP, and in each of those RuBP molecules you have five carbons. So these three molecules of RuBP are going to come in, and then they're going to attach with three carbon dioxide molecules, each containing one carbon, and the enzyme that does carbon fixation there is that enzyme called rubisco. And then you form the first stable product of the Calvin cycle called PGA. The next step is the reduction step, where you take PGA, and PGA is going to be converted to our molecule that will become the sugar. And in order to go through that step, you need to have an input of energy. So 6 ATP molecules are going to come in, and those were generated during the light reactions of photosynthesis. And then you also have 6 NADPH molecules coming in. And those 6 NADPH molecules are going to be oxidized, and PGA will be reduced to form glyceraldehyde 3-phosphate, or G3P, also known as PGAL. So phase 2 here of the Calvin cycle is called the reduction step. The last step is the formation again of the regeneration of the carbon dioxide acceptor called RuBP. So of those 6 3-carbon molecules, one of those G3P molecules comes off, and it's going to be part of the formation of a sugar molecule. The other 5 continue back in and are reconverted when 3 ATP molecules come in, and they're going to be reconverted back into that RUBP, which is the starting molecule for carbon fixation. Now, we only have one 3-carbon G3P molecule as a sugar output for this one turn. But we know that the sugar molecule has 6 carbons. So we need to do this a second time in order to get out 6 carbons to go on and make our sugar. So again, those 3,5-carbon RUBP molecules are going to be fixed to 3 carbon dioxide molecules, each containing 1 carbon, using the enzyme Rubisco. And you're going to form the first stable product of 6 molecules of PGA, each of those having 3 carbons. That's carbon fixation. The next step is the reduction stage, where you're going to have those 6 3-carbon PGA molecules be converted to 6 3-carbon G3P molecules after 6 ATP come in and 6 NADPH. Now, those 6 G3P molecules, one of them will be lost to go and form the sugar. So we have another 3-carbon G3P molecule coming out of the cycle. And the other 5 will then continue back in and be reconverted to RUBP when 3 ATP molecules come in and supply their energy. And then again, you have reformed that carbon acceptor molecule. Now, after the second turn, so this occurs twice, after 2 turns of the Calvin cycle taking place in the stroma, you now have enough carbons to go on and make glucose and other organic compounds. Next, now we're going to look at the alternative pathways or alternative mechanisms for photosynthesis. So dehydration is a problem for plants and sometimes requires trade-offs with other metabolic processes, especially photosynthesis. On hot, dry days, plants close stomata, which conserves water, but also limits photosynthesis because you need to have that gas exchange occurring. So the closing of the stoma reduces access to carbon dioxide and causes oxygen gas to build up. These conditions favor a seemingly wasteful process called photorespiration. So we have, typically, if we go back here, we did the C3 cycle. Well, now we're going to look at C4 and CAM photosynthesis. So in most plants, C3, 3-carbon plants, the initial fixation of carbon dioxide via Rubisco, that's the enzyme, forms a 3-carbon compound. That 3-carbon compound was PGA. In photorespiration, Rubisco adds oxygen to the Calvin cycle instead of carbon dioxide. So now we're going to see oxygen coming into play here in place of carbon dioxide. And photorespiration consumes the oxygen and organic fuel and releases carbon dioxide without producing ATP or sugar. Photorespiration may be an evolutionary relic because Rubisco first evolved at a time when the atmosphere had far less oxygen and more carbon dioxide. So here we could see Rubisco carboxylase or oxygenase. So that's the name of the enzyme here. So we could see that Rubisco oxygenase and came later because at first that initial atmospheric conditions were built up with carbon dioxide gas. And if you think of all the volcanic activity of our very young Earth, carbon dioxide gas is being spewed into the atmosphere. So Rubisco carboxylase would have been the enzyme used for fixation. So in many plants, photorespiration is a problem because on a hot, dry day, it can drain as much as 50% of the carbon fixed by the Calvin cycle. So C4 plants, how do they do this? In C4 plants, they minimize the cost of photorespiration by incorporating carbon dioxide into a 4-carbon compound in mesophyll cells. Then these 4-carbon compounds are exported to the bundle sheath cells where they release carbon dioxide that is then used in the Calvin cycle. Now, this is a spatial separation of what's going on here. So if you look, here you can see the mesophyll cells Down here you can see bundle sheath cells. So this carbon dioxide is coming into the mesophyll cells, and an enzyme called PEP carboxylase is going to carbon fix that to a 4-carbon compound called oxaloacetate. And you'll see this compound later. And then oxaloacetate is going to form malate, and then that's going to form pyruvate. Now we're down in the bundle sheath cells. So when you go from malate to pyruvate, you're going to lose a carbon. And that carbon lost is going to be that carbon dioxide molecule that is going to feed into the Calvin cycle to make the sugar that will eventually end up in the vascular tissue, the phloem tissue. So here we have a spatial separation, 2 different areas of the plant cells, the mesophyll cells and the bundle sheath, where this process is going to occur. In CAM photosynthesis, the plants open their stomata at night and incorporate carbon dioxide into organic acids. During the day, the stomata close, and carbon dioxide is released from organic acids and used in the Calvin cycle. So here you can see in CAM photosynthesis, carbon dioxide is incorporated into that 4-carbon organic acid. That would be carbon fixation. So here is the C4 plant. A typical C4 plant would be sugarcane. Here is a CAM plant. So pineapples are good CAM plants. So at night, CO2 comes in, it's fixed into organic acid, and then in the day, when the stomates are closed, the organic acid will release carbon dioxide into the Calvin cycle, where then the sugar will be made. So where the C4 pathway was a spatial separation, in a CAM pathway, it's a temporal separation. So during the day, the stomates are closed, and at night, the stomates open up when it's cooler for these plants, and they take in carbon dioxide gas and fix it into a 4-carbon organic acid that will later in the day release that carbon dioxide molecule into the Calvin cycle. So the overall importance of photosynthesis: the energy entering chloroplast as sunlight gets stored as chemical energy in organic compounds. Sugar made in the chloroplast supplies chemical energy and carbon skeletons to synthesize the organic molecules of cells. And then in addition to food production, photosynthesis produces the oxygen in our atmosphere. That's it for the chapter.